And this is where we left off. We're ready to move on to the next slide. We talked about carbon dioxide transport, and again, most of it is a bicarbonate ion. There is a enzyme that's important, and so it's important to get in your head that carbonic anhydrase in red blood cell promotes useful changes in partial pressure of carbon dioxide. So what you have in this formula, carbonic anhydrase is listed as the CA. It's a enzyme that facilitates this reaction. If you have high partial pressure of carbon dioxide, then water and carbon dioxide is going to be turned into the carbonic amino hemoglobin, I guess. No, yeah, that actually, no, that's not what that is. That's a molecule with carbon dioxide in it, and that then can turn into the bicarbonate ion plus hydrogen. Sorry, I don't exactly remember what the name of H2CO3 is. It's a temporary molecule. I know we put it in there. Sorry, I don't remember that. If carbon dioxide is low, then the reaction is going to go the other direction. What you're going to do is take that attached form of carbon dioxide and put it into the plasma as carbon dioxide and water. One of the reasons why this is important is, first of all, you want to remember this enzyme, carbonic anhydrase, but secondly, remember that throughout the whole process, this is happening in a passive way. So it's happening because oxygen is more concentrated in one spot than another. If in fact these partial pressures of gases cannot be controlled somewhere in terms of their percentage of pressure, so how much of it is actually in that dissolved plasma liquid, then that piece where the oxygen drops off and the CO2 is absorbed into the bloodstream and vice versa at the lungs is not going to happen. So this is important not only with regard to oxygen and its ability to be stored on those red blood cells. That oxygen that's stored on the red blood cells does not come into factor with regard to the partial pressure of oxygen in the plasma, in the blood plasma itself. It's the partial pressure of the oxygen that's dissolved in the liquid, not being held by those red blood cells. And then with carbon dioxide, which carbon dioxide is the bigger driver of the shift left or right, if it is dissolved in the plasma as carbon dioxide, it increases the partial pressure. So what happens here is it's a way for the red blood cells to actually control the partial pressure of carbon dioxide. And with this, and we'll see this on, I think, the next slide, in order for the red blood cells to do this and remain their own electroneutrality, what happens is that bicarbonate ion is temporarily expelled out of the red blood cell, and that's a negative ion. So what the red blood cell does is then takes in a chloride ion. Chloride ions are always floating around. It's one of your electrolytes. It's one reason why you need chloride, and so this tends to be what's called the chloride shift. So chloride ions help maintain the electroneutrality, meaning one negative, one out, one negative in. So negative is in equilibrium. The solutes may not be in equilibrium, but the negative is in equilibrium. From red blood cells diffuses into the plasma as carbon or bicarbonate ion. Red blood cells become more positive as that ion moves out, and then the chloride is attracted in. You can even say it's co-transport. The hydrogen release buffered by a combination with deoxyhemoglobin then is buffered by this bicarbonate ion. Reverse is then going to happen in the capillaries. And then here's the picture. This is where I wanted to get to. So on the left, you have this red blood cell. It is actually picking up CO2 from the cell itself, cell metabolism, that waste product, picking it up. It's turning it into this, with the help of carbonic anhydrase, it's turning it into this temporary molecule H2CO3. Remember, not too many of these are stored in red blood cells. Most of this, this is a passageway. So it's a process. This H2CO3 is more of a temporary molecule. So then the hydrogen can bind to the hemoglobin, and the bicarbonate ion can move on out of the red blood cell, can stay dissolved in the plasma. And that's when the chloride goes in. That's when you have that chloride shift, that shift from bicarbonate ion to chloride. And then what happens is this red blood cell goes along in the systemic circulation, goes back to the capillary beds that are around the alveoli. And when they get there, then what's going to happen now is the bicarbonate ion is going to go back into the red blood cell. It's going to mix with the hydrogen, or with the hydrogen ion that's going to come off the hemoglobin. Chloride is going to shift out, and then the red blood cell is going to release carbon dioxide and water into the plasma, and then the carbon dioxide is going to diffuse across down a concentration gradient into the alveolus. So this is the process by which carbon dioxide is picked up by all the cells in your body, transferred or transported in blood, and then when it gets to the alveolar area is able to be dropped off and then expelled out in exhalation. In terms of regulation of transport, there are, as I said, there's a large reservoir of oxygen on hemoglobin. So the body doesn't tend to look at oxygen levels, which is kind of surprising the first time you hear that, but it makes sense. You have plenty. So what you are looking at, what your body's looking at in its infinite wisdom, via chemoreceptors are changes in carbon dioxide, the partial pressure of carbon dioxide. This is sensed through pH. Carbon dioxide can lower the pH or raise the pH, whichever the scenario might be. And then ventilation will be adjusted because you breathe out carbon dioxide. Be adjusted to maintain arterial CO2 levels at about 40 milligrams of mercury. These chemoreceptors are located in the brain, and also the aortic arch, as well as throughout arteries throughout the whole body. And this material does tend to be asked again, that it's carbon dioxide that is really the driver of your breath rate and depth, and it's these chemoreceptors that are constantly looking at blood, particularly as it's going out of the heart throughout the whole arterial system. And if we look at the images over on the right, you have hypoventilation, that's less than 12 per minute. That's going to increase your carbon dioxide. It's going to increase the hydrogen ions, and it's going to decrease your partial pressure of oxygen. Chemoreceptors are going to pick that up, respond, send a sensory impulse to the respiratory control center of the brain, and the brain's going to say, we need to increase ventilation, and that will be a negative feedback to basically bring things back to normal. Hyperventilation, which probably we're more familiar with, that's going to decrease, so you're breathing out too much CO2, decrease carbon dioxide partial pressure, decrease hydrogen ions, but increase oxygen partial pressure. That's going to be picked up, and ventilation will be decreased. Your respiratory rate will be decreased. So you see both of those as a negative feedback, and it actually is happening quite often in your body. The neural centers for breathing, involuntary are in the medulla and the pons. Voluntary is cortex, and breathing is an involuntary process until you want to control your breath. So just again, think of somebody who plays an instrument, or somebody who sings, etc., or even just somebody who talks. We have voluntary control of our breathing as well as involuntary control. That involuntary control comes from our brain stem via the medulla and the pons. And then you have in this image, and I'm trying to get all this done in this one, but you have this image of increase or decrease of oxygen or carbon dioxide, and how that will cause a positive or negative effect with regard to rate and rhythm. Things that cause a positive effect would be increase of CO2, increase of hydrogen ions, and that particularly would be in the periphery, where the peripheral receptors. You have an increase in maybe an irritant. There are involuntary breath, like coughing, etc., if you have an irritant, or if you have a muscle need. These would all cause, it would send information to the brainstem and shift the rate with which you are taking a breath in. Couple of things to remember. It's the phrenic nerve that is instigates that starts the diaphragm. The phrenic nerve innervates the diaphragm and the intercostal muscles. Rate and depth are in the medulla. So the medulla oblongata is responsible for rate and depth, and smoothing out the rate is the pons. So those two areas work together to control your rate and depth. So the rate and depth are controlled by the medulla, smoothed out by the pons. Respiratory rate again is around 12. It's around 12 to 15, but around 12 we hit it at that. And then that rate can either be higher or lower, and then we do have hypernia and hypoernia. Things that affect your depth, your body temperature and rate, exercise, talking, and coughing. As I said, if in fact you need to take air in because you're coughing. Volition, you have conscious control, and emotional factors can affect your rate and depth. So certain physical factors can increase or decrease, so body temperature, exercise, talking, coughing. And also if you have an irritant, that is going to cause a involuntary, I guess, maybe shift in the rate and depth. And as well as emotional factors. You've probably seen this in people who they just can't take a breath in because of emotional things that have happened. And that's the end. I didn't know that was the last one. So anyway, we will move on to the next chapter.